All right, so we're going to talk about bleeding and the stoppage of bleeding. Welcome back to part 3 of blood. So we're talking about hemostasis. Okay, so this is a big, messy diagram. And we have some steps, so we're going to look at the 3 basic steps when there is a breach in the wall of a blood vessel. So we are going to have vascular spasm, platelet plug formation, and true coagulation. All this mess over here is actually the coagulation cascade. We're going to talk about the intrinsic pathway and then the extrinsic pathway. This part, I'm not really going to ask you to reproduce that, so don't stress and just kind of hang there with me. We can do this. All right, so that word heme, I said that always means blood. Stasis means to stop. So hemostasis is a stoppage of bleeding, and there's 3 overlapping phases to this. The first one, blood vascular vessel spasm, and it sounds just like that. It's a spasm, and we'll look at it in a second, but there's smooth muscle that's basically going to constrict. If you think about it, if there's a breach in the vessel wall, we want to restrict blood flow so we don't lose that much. So that's the function of that. The next part is what's called platelet plug formation, and a platelet plug is not actually a blood clot. So those thrombocytes, those little platelets, they're going to help. They're going to form this temporary seal, but then it's not till the 3rd step, the actual blood clotting or blood coagulation, where we are going to get some protein fibers, fibrin, that are going to stabilize this blood clot and stop the bleeding. There's a lot of proteins involved in this called clotting factors. And so if there is an absence of a clotting factor or there's failure along the way, that's when you can get hemorrhage. So that's excessive bleeding. So that first step, the vascular spasm. So this is a view of the cross-section of a vessel. We're going to learn this later, but it's arteries, arterioles, capillaries, venules, veins. The middle layer of vessels is smooth muscle, and so that's called the tunica media. But this smooth muscle, when there is damage, we are going to get that constriction. And like I said, that just kind of makes sense. That's just going to reduce the amount of blood that's going to be lost. It's going to be triggered by pain receptors. So, and then from the endothelial serotonin, and then platelets, which we're going to add next, they're also going to release factors like prostaglandins that are going to further cause that vascular spasm. Okay, so let's take a look.
In a first step towards clot formation, platelets are recruited to the site of vessel injury by now exposed molecules of the vessel wall, such as collagen and von Willebrand factor. This factor mediates the linking of platelets to collagen via a specific receptor in the platelet membrane. The resulting change of shape of the platelet from its resting state into the dendritic form indicates activation. The activated platelet in turn releases prothrombotic molecules such as adenosine diphosphate, ADP. By binding to its receptors, ADP induces aggregation. And recruits further platelets to the site. Thromboxane is another important mediator of platelet activation and aggregation. Under its influence, the platelets cross-link with each other. These interlocking mechanisms cause platelet activation to snowball. The clot grows rapidly. Activated platelets also trigger the coagulation cascade and thus the formation of thrombin. Thrombin, in turn, stimulates platelet activation even further, a continuous feedback loop. Additionally, thrombin induces the formation of fibrin for the mesh stabilizing the clot. The self-reinforcing process of platelet activation, crucial in the formation of blood clots, is an obvious therapeutic target in conditions caused by inappropriately triggered blood coagulation.
When there is a break in a blood vessel wall, then there are exposed collagen fibers. So that is what the platelets are going to stick to. Once they stick, then they get activated, and it was shown in that, I don't know what's wrong with my mouth, sorry. Um, in that YouTube, that's when they kind of, got little appendages and they change shape. So when they become activated, they are going to release what's inside of them. And remember, they're not true cells. They're just going to be these little packets of chemicals. So when they release them, ADP was a big one, platelet factor 3, platelet-derived growth factor. All these stages are happening kind of at once and they're overlapping. But if we have a break in a vessel wall, we also need to have restoration. We need that vessel to be fixed. So we are already starting that growth factor. We're going to stimulate that endothelium to grow back. Like I said, we get aggregation. Also, we're going to turn on the clotting pathways, and that platelet-derived growth factor is going to initiate healing. And then two, I don't know if you remember back from AP1, there was only two examples of positive feedback. One was that childbirth we talked about in the endocrine chapter, and this is platelets. When they get activated, they activate more platelets, and they call them to aggregate and clump together. They call them to that site of damage. So they have to adhere, they get activated, and they aggregate and form that platelet plug. So things that can go wrong with this formation of the platelet plug. There's something called von Willebrand disease. It is the most common bleeding disorder. It is either an absence or a reduced function of that factor. And just like we saw, we're not up to hemophilia yet. We're not really talking about forming a true clot. We're just talking about forming that platelet plug, that temporary soft clot, if you will. And so von Willebrand factor, it helps the platelets stick together. So if that's missing, they just don't stick together as well as they should. It means there's going to be a delayed blood clotting time. It's genetic. There's 3 different types. Some are more serious than others. They don't usually cause death. They cause bleeding symptoms, so definitely in women, extra heavy menstrual flow. If there's any sort of minor injury, you get way more bleeding, excessive bleeding of the gums in both genders. 2, this is a problem if someone's going to have surgery or even if they have tooth extraction. But it, like I said, is the number one bleeding disorder. So von Willebrand disease. So that lacking of that factor. So we're also going to be talking about preventing inappropriate blood clotting. So anticoagulants, so anti means against, coagulant, so against blood clot formation. Since one of the major steps in this process is formation of that platelet plug, what we don't want to see is blood clots inside of healthy vessels. So what healthy vessels do is that endothelium, so the cells lining a blood vessel, they're going to release anticoagulants, so things like nitric oxide and prostacyclin. I don't know if you know anyone that is taking aspirin. That is also an example of an anticoagulant. It inhibits platelet plug formation and actually inhibits the production of thromboxane by the platelets. There was a very large study that was done where half of the study group just took this small therapeutic dose of aspirin every day compared to the group that did not. And just taking those small little baby aspirin every day reduces the risk of heart attack by 50%. So it's a really cheap and expensive thing to do. Everyone 50 and older should do it. If you have someone in that age group that you love, just go buy them some baby aspirin. But it just helps to prevent that platelet plug formation. When we have inappropriate clotting in the vasculature of the heart, that's when you have a heart attack, and inappropriate blood clotting of the vessels going to the brain, that's when you have a stroke. So it helps prevent that. Okay, so we have vascular spasm, platelet plug formation. So our third and final step is blood coagulation. So that's when a true clot forms. And so that's what this is showing right here. This is a fibrin meshwork. So these fibrin strands are going to reinforce that platelet plug. So blood is being transformed from a liquid to a gel. And this is going to be through a series of reactions. So you can think of a clotting cascade. What I want you to know about these clotting factors, the majority of them are going to be made by the liver. They were given Roman numerals in the order they were discovered, not in the order that they act. So 1 through 13. Think about what needs to occur. Your body always has to be ready to form a blood clot in case you get cut. But you don't want it to form an inappropriate clot. So these proteins are going to be circulating in an inactive form. but they're ready. Other things I want you to keep in mind, because it can come up clinically, vitamin K and calcium are needed for this blood clotting process. So there are two pathways that lead to the same place. So they're all going to, both these pathways lead to prothrombin activator and what's called the common pathway. So we have the extrinsic pathway, and that word extrinsic means something from outside. So the extrinsic pathway, to be honest, this is like normal blood clotting, like you think of when you cut yourself. It involves factors from outside the blood, so it can involve tissue factors. The intrinsic pathway involves factors only inside of the blood. So yes, so we're going to look at the extrinsic pathway first. So once we get to clotting factor 10 and activated clotting factor 10, then we're going to get to this common pathway. So no, I'm not going to ask you these steps in between. But what I do want you to know, look at this extrinsic pathway. So we have trauma to extravascular cells. Okay, so you've had an injury. We are going to get tissue factor, so from outside of the blood. So that's going to be the trigger for this branch of blood clotting. Notice it has fewer steps. It is going to be faster. And so it takes seconds to a few minutes for a normal blood clot to form. And so that's the extrinsic pathway.
Hemostasis is the normal physiological response to limit blood loss after vascular injury. Blood clots are made up of activated platelets, red blood cells and fibrin, which hold the clot together. The process is mediated by around 30 proteins, which are known as clotting factors. Platelet activation and aggregation begins when vascular injury exposes collagen and von Willebrand factor from within the vessel wall to the blood. Platelets adhere to these proteins and become partially activated, changing shape and releasing signaling molecules which recruit more platelets to the site. At the same time, a protein called tissue factor triggers initiation of the coagulation cascade, the process that leads to full activation of platelets and the generation of fibrin and clot formation. Vascular injury causes tissue factor on the outside of cells in the vessel wall to come into contact with factor 7A and factor 10A in the blood, leading to the production of a small amount of thrombin. This thrombin fully activates platelets and triggers a cascade of clotting factors that activate each other, culminating in factor 10A mediated generation of a larger burst of thrombin production on the platelet surface. One molecule of factor 10A leads to the formation of 1,000 molecules of thrombin. Thrombin converts the soluble protein fibrinogen into insoluble fibrin strands at the site of vessel injury, while at the same time activating factor 13 protein to cross-link the fibers. The fibrin mesh stabilizes the clot and traps more platelets and red blood cells, facilitating its growth. In normal physiological conditions, the process of clot breakdown or fibrinolysis begins as the structural integrity of the vessel wall is restored. Endothelial cells begin to secrete a protein called tissue plasminogen activator, which activates the protease plasmin to degrade fibrin, dismantling the structure of the clot and restoring the integrity of the vessel.
Okay, so that is a really good YouTube. I'm going to put this link for you in your checklist. That's not a bad one to watch again at your leisure. So the extrinsic pathway, that is when we get blood coagulation with tissue damage because we have that release of that tissue factor. Blood, if you collect it in a test tube or in a capillary tube without an anticoagulant, so something like heparin, EDTA. We also said that aspirin and warfarin, I haven't brought that up yet, but there's also other anticoagulants, but usually what's going to be used clinically, heparin, EDTA, citrate is another one. So if you take blood and just leave it in a tube. You could spin it. We already said we have plasma and blood. But what's going to happen over time through only this intrinsic pathway, so it's going to take longer, so it can take up to 15 minutes to 20 minutes, is that you will get blood clot formation inside of a tube. And so it's just going to be the positively charged walls of either of the glass vessel or the tube that's going to cause this blood clotting strictly through that intrinsic blood clotting cascade. So when you have the formed elements and you have all of the clotting proteins that have formed this clot, what you're left with is that fluid portion. So plasma minus blood clotting proteins is called serum. So that's where you get serum from. So let's take a look at, like I said, this side. So the intrinsic pathway. It is the pathway that they're happening at the same time. I don't think they're completely separate. So even when it's inside the body and there is a cut, this pathway is overlapping with the extrinsic pathway. There are more clotting factors involved, so it's going to be slower. If, like I said, if there's blood that's drawn and collected in a tube without an anticoagulant, then it's only the intrinsic pathway. Hopefully I didn't make that too confusing. But the big thing, intrinsic, all from the factors that are just within the blood, so without that tissue factor. And the big thing is it's slower, and both extrinsic and intrinsic are going to lead to this common pathway. Eventually we're going to get fibrin and that true blood clot. And we're going to look at that common pathway in a second. So look at all of these clotting factors, so they each had Roman numerals. So there's a gene for each of these, and sometimes someone is missing that gene or it is a non-functional gene. And so if they're missing one of these clotting factors, that's when they have hemophilia. So that's when they can't form that blood clot. And there's different kinds, hemophilia A, B, and C, and it just depends on which clotting factor they're missing. So A, they're missing 8, B, they're missing 9, and C, they're missing 11. No, I'm not going to ask you that. But that's where you get the different types of hemophilia. Most of these genes, strangely enough, are on the X chromosome. So females have 2 X's, so they would have to inherit 2 bad copies of that gene to be hemophiliac. And males, since they just get 1 bad copy, then they'll have hemophilia. So that's why the majority of hemophiliacs are going to be male. So we're going to take a moment and talk about inappropriate blood clotting. So this is clotting with no tissue damage inside of a vessel. So these are not good. Like I said, this is the cause of heart attacks if it's the coronary circulation. Also, if you've heard of mini strokes or TIAs, so transient ischemic attack, full-blown strokes, so cerebrovascular accidents, and also pulmonary embolism we're going to look at in a second. So there's some terminology. So if you've heard the term thrombus, that is a blood clot inside of a vessel. So inappropriate blood clotting. If that thrombus breaks off or dislodges and travels in the bloodstream, then it's called an embolus. When it gets lodged and blocks a vessel, then it's an embolism. So those are just some terminologies. So different types of strokes. It could be a blood clot that forms inside the brain that would cause a thrombotic stroke. Think about it, embolytic stroke. Embolism, that word actually just means wedge, and so that would be a fatty plaque from somewhere else or a blood clot that has come from somewhere else and then wedges itself inside the circulation inside the brain. And then 2, there's also red stroke. That's when someone bleeds inside their brain, so an aneurysm. So let's go ahead and look at pulmonary embolism. Whenever you see pulmonary, it always refers to lung, and so that's when a clot lodges inside the lungs. And so breathing problems, it often leads to death. It can be drug-related, environmental, or genetic causes. Often when lack of movement, so on long airplane rides, you can get deep vein thrombosis that then turns into a pulmonary embolism. So let's go ahead and watch.
Pathological thrombi can be extremely serious, causing complications such as stroke, pulmonary embolism, or heart attack. 2 different types of thrombi can form, arterial thrombi and venous thrombi. Here we will focus on venous thrombosis. In the slow-moving blood of the venous system, changes in blood composition, a reduction in blood flow, or changes to the vessel wall can result in the formation of fibrin-rich clots. These clots form through activation of the coagulation cascade, culminating in the factor Xa mediated generation of thrombin and the thrombin mediated cleavage of soluble fibrinogen to insoluble fibrin. Fibrin forms long strands which are cross-linked to form a mesh that traps passing red blood cells and platelets. Over time, these build up to form a clot. If this kind of clot forms in the large veins of the leg, it is termed deep vein thrombosis or DVT. Alternatively, a part of the clot may break away and be carried in the bloodstream to the lung. If it blocks one of the arteries in the lung, this is termed a pulmonary embolism and can be fatal. In either case, clot resolution is vital because of the key role of the coagulation cascade in the formation of venous clots. Anticoagulants, which target specific proteins within the cascade, are currently the guideline-recommended treatment for acute DVT and pulmonary embolism in most patients.
Okay, so let's just recap. Why does intravascular clotting happen? Sometimes the endothelium is just roughed up instead of smooth. As a person ages, it just happens over time. So atherosclerosis is that fatty plaque buildup. So when blood flows, you've got a narrowing here. Blood can then circulate or brew in those areas, and platelets can bump up against those walls and they can get activated. The other thing that can happen is often these plaques will rupture and then they form an embolism, so then they move. Other things too, just like when they call the deep vein thrombosis, sometimes the blood moves very slowly. And so if it moves slowly, that means you get an aggregation of those blood clotting proteins. And like I said, it just kind of causes, it's only a couple platelets that need to get activated. They're going to start and kick off that blood coagulation cascade. And so we just looked at deep vein thrombosis. We're going to go into talking about the heart. So atrial fibrillation, also when the top chambers of the heart fibrillate, that just, whenever blood's not moving, it's a bad thing, and you increase your risk of intravascular clotting or inappropriate blood clotting. Okay, so both the extrinsic pathway and the intrinsic pathway are going to meet right here at prothrombin activator. And it's, once again, these are enzymatic cascades. So where one protein is going to activate another protein in the sequence, and then that protein is going to activate another protein. So we are going to have prothrombin activator. Pro, like a prologue, is before. So before thrombin, you have to have this activator. And circulating in its inactive form is prothrombin, so before thrombin. You also need calcium. Then prothrombin activator is going to cause the cleavage of some amino acids, so we get inactive prothrombin to active thrombin. When thrombin is active, what its job is, it is going to convert the soluble blood protein fibrinogen. So remember I said genesis, the beginning of fibrin. So this is the inactive soluble form. Once again, it requires the presence of calcium. Then we get fibrin. And so that's the fibers that make that meshwork of that blood clot. So fibrinogen is a soluble protein. Fibrin is an insoluble protein. Vitamin K is needed for the production of prothrombin. And warfarin or Coumadin is another name. This is an anticoagulant. Often if someone has DVT or if they have atrial fibrillation or heart issues, they may be prescribed these anticoagulants to prevent inappropriate blood clotting. And so it interferes with formation of prothrombin. So we talked about, we have vascular spasm, platelet plug formation, and now we have a true blood clot. So the clot forms, we have this fibrin meshwork, and it traps the red blood cells and the platelets. And we are also now, we need to set the stage for repair. We want the walls of the vessel to be brought together, and then we need that vessel to repair itself. So we're going to get clot retraction, and it's actually those platelets that are going to retract and pull those torn edges of the vessels together. Through the intrinsic pathway inside a test tube, it is going to be that platelet retraction that is actually going to squeeze the serum out of that blood clot. So that when I showed you that other picture, that's what you were actually looking at. We already have growth factors that have been released by the platelets. We talked about those. That's going to stimulate mitosis and the growth of those cells that make up that vessel wall. The endothelium is also, when it gets ruptured, releases vascular endothelial growth factors. So we're trying to repair. As that blood vessel repairs, though, we want that clot to dissolve. So there's a process for that as well. So that's called fibrinolysis. In the blood, there's a protein in inactive form made by the liver. It's magical. Plasminogen, so the genesis of plasmin. There's also plasminogen activator. So that protein is going to convert plasminogen to plasmin. Plasmin is a clot dissolver or a clot buster, if you want to think of it that way, and that's going to dissolve that clot. You don't want that clot to last forever. So, tissue plasminogen activator, it has been cloned, it's available pharmaceutically. So it is a clot busting drug, so TPA. So if someone comes in and a stroke has occurred or a heart attack is in progress, then they will administer tissue plasminogen activator. And so it's going to activate plasmin. It's going to then, since plasmin is a fibrinolytic, so it's going to cut up fibrin, or a thrombolytic, a clot-busting drug. And so the hope is that blood flow will be restored. That way, if it was a heart attack, it would reduce the damage to the myocardium, to the cardiac muscle. And if it was in the brain, it would restore blood flow going to those neurons, so there would be less brain damage. And so TPA, tissue plasminogen activator, is going to be given through an IV to break up clots during heart attacks and ischemic strokes. Okay, I'm going to wrap this one up here. Come back for the last installment of Blood, so part 4. Enjoy the rest of your day.